My name is Robert Hoffman. I'm the founder of AntiCAncer Inc. more than 35 years ago and the founder of AntiCAncer Japan. Today I would like to talk to you about methylation, especially in disease, including cancer. What is methylation? Methylation is the transfer of methyl groups from the amino acid methionine to other molecules, such as DNA, proteins, lipids. But methionine itself cannot donate its methyl group. Methionine must be activated by being converted to S-adenosyl methionine by the enzyme methionine-adenosyl transferase. S-adenosyl methionine, sometimes called SAM for short, is the universal methyl donor in the cell. The cell has many enzymes called methyl transferases, which use S-adenosyl methionine and transfer the methyl group from methionine as part of S-adenosyl methionine to other molecules, as mentioned before, such as DNA, and proteins, and lipids. When a molecule gets methylated, its function can change dramatically. For example, if DNA is methylated at the appropriate sites, always at cytosines, it can change the expression of DNA, whether a gene will be expressed or not. This is a very crucial modification of DNA. It also turns out that the proteins which surround DNA, called histones, also are methylated, and the methylation of histones can also dramatically change whether a gene is expressed or not, or the degree of expression of that gene. In cancer, it seems that the methylation process is greatly altered, and the rate of methylation goes up, making the cells methionine addicts. They require much more methionine, but the methylation reactions that occur in the cancer cell seem to be very different than what occurs in normal cells. And these changes in DNA methylation and histone methylation that take place in cancer alter the expression maybe of thousands of genes. They alter the genetic program, the readout of the genetic program in cells, making the cells cancerous. This seems to be the major change that takes place in a cancer. And paradoxically, the overall methylation of DNA seems to get lowered in cancer, even though there are many transmethylation reactions occurring at very high rates. But DNA itself seems to become hypomethylated in cancer, a discovery we made in human cells back in 1982. And this greatly alters the genetic readout, the genetic program of the cancer cell, with thousands of genes perhaps changing their activity, changing the phenotype of the cell into one that's cancerous. The cells are, as mentioned, methionine addicts in cancer. They require much more methionine than normal cells, and they can be treated by methionine restriction, either by diet or much more effectively by an enzyme called methioninase that can be administered orally. Methylation changes can affect other processes in the body. It seems that if the level of methionine is lowered, the proclivity toward obesity and diabetes is decreased. So again, probably the depletion of methionine by methioninase or diet preventing obesity or diabetes involves changes in the genetic program readout in the cells. It is also possible that low levels of methionine and lower levels of methylation in the cell can greatly extend the normal healthy lifespan This is a very exciting opportunity that we hope to be able to exploit in the future. Thank you very much for listening.